Everybody. So now a very different topic here. So adipose tissue, the big daddy of the liver. To tell you a story, and stories are for kids who are sitting on the front rather than the people on the back side. An old story, which means I am taking you in time to the Ordovician period, around 480 million years ago, when we all were cockroaches. And my question, does a cockroach have a liver? Does it has immune cells or bone marrow? Ask you a question, how many of you have dissected a cockroach? Different. Yeah. So if you dissect a cockroach, you will see that the first thing you will notice is that it is full of fat bodies. The very cockroach does not have a liver, it does not have a bone marrow or immune cells, but it has fat bodies which is equal to the adipose tissue, which also does the function of a liver, immune cells, and adipose tissue. Now you will ask me, hey Sanal, thanks for your stories. Does it has any does your stories have any clinical relevance, or do you have any proof for that? Pediatric residents will immediately recognize this. Can you please tell me what is this image? Sleeping. Or they are figuring out how to use the mic. That also I don't know. But maybe this is a blueberry muffin baby, which is a consequence of congenital rubella syndrome. Now my next question is that I am giving you a couple of slides, glass slides, and if you press them against these spots, will they blanch or not? There is no, because they represent subcutaneous hematopoiesis. They are hematopoietic foci, ectopic hematopoietic foci. They arise from the dermis, the adipose tissue which is beneath it. So this is a proof. And if any of you come across such babies, I need a tissue biopsy and it will be a great help for me. It means a lot for me, you or any of your colleagues across India. Or maybe from India I may not get it because everybody is vaccinated. I may have to go to Pakistan or Trumbistan. So now the part 2, adipose tissue, the jack of all trades. Not just fat, it is an endocrine organ. It produces key hormones like leptin, adiponectin. It stores energy. It burns fat. It prevents lipotoxicity and prevents pancreatitis. Perhaps you know that one of the key deviate. So it keeps you warm, it's an insulator, it cushions your butts and heels, and stores vitamins, and it's an immunological organ, and it gives you figure, softness, and suppleness. Think about your curves, forget weight, and it is the waist-hip ratio which is more important than the BMI or total body weight. Waist circumference is an indicator of the visceral obesity, and a lot more. Now coming to part 3. What we seek is often found in what we stumble upon. Perhaps many of you know that my projects are funded for gene therapy rather than any adipose tissue research. And the project which we got funded was for hemophilia gene therapy. And while we were trying, our vectors, we purchased an antibody to prove that hemophilia gene is being expressed. So when you try an antibody, you always need a positive control and a negative control. The negative control says that the antibody is not aberrantly reacting or non-specifically reacting. So we used the liver tissue as a positive control, and adipose tissue we thought as a negative control. And to our surprise, the adipose tissue gave us positive signals. As you can see here, very clear positivity probably it's not well focused here, but you can see in cirrhosis, in both healthy and cirrhosis, it showed positivity. These brown areas are where factor VIII is positive. That means we thought that initially that it may be a mistake, so we purchased another lot of antibodies, which again we got the same result. So we were taken by surprise. So we thought that, okay, probably anyway antibodies can be non-specific. So what we'll do then? We'll go for a Western blot, because when you do a Western blot, it's not only about specificity, but also you are testing the molecular weight for that entity. And it showed clearly that factor VIII, a 330 kilodalton protein is present there. Then science is all about being critical. We need to question. What if somebody argues that, what if factor VIII is present in adipose tissue, but it is not produced there? It is just produced by liver and getting deposited in adipose tissue. That is when we did a real-time PCR, where you are not looking for the protein synthesized, but the RNA transcripts, the messenger RNA, which codes for the protein. So if you isolate that from the cells, it clearly tells that it is produced in that tissue. So as you can clearly see here, in cirrhosis, the mRNA level for factor VIII is way higher, and it is clearly present. And in healthy, it is there compared to the GAPDH, and in cirrhosis patients, its levels are way higher. If factor VIII is produced in adipose tissue, which cells in adipose tissue are producing factor VIII? Because as liver is not full of hepatocytes, only about 80% or more, slightly more than that are hepatocytes. So rest are composed by other type of cells. So same is true for adipocytes. So as you can see here, we did a FACS. The FACS tells you clearly that the CD31 positive cells in the adipose tissue compartment are expressing more of factor VIII, and that is way more in the cirrhosis patients compared to the healthy. Again, further, as you can see here, CD34, which is a marker of hematopoietic stem cells or even primitive endothelial cells, those cells are also quite high. You see this corner, very high in adipose tissue from cirrhosis patients. So this is the figure here. See the CD34 cells, which are way higher, they are present in adipose tissue. Not only they are present, their numbers are way high in people with cirrhosis. This is a cirrhosis patient's adipose tissue. This is a FACS. Now, what about liver? So, if you can ask me, can you give me a comparison of adipose tissue versus liver? This is the picture here. This is factor VIII in adipose tissue, healthy adipose tissue, and this is liver. And this is adipose tissue from cirrhosis, and this is liver tissue. As you can see here, factor VIII is expressed more in adipose tissue in cirrhosis patients, and definitely even more it is expressed in liver from a cirrhosis patient. And what about von Willebrand factor? Why? Because von Willebrand factor carries factor VIII and protects it. And as you can clearly see here, the von Willebrand factor level is also high in cirrhosis as well as in liver. So what is behind the coagulation profile or abnormal coagulation profile in cirrhosis? As you can see here, this is the ratio of liver versus adipose tissue with reference for factor 8 and von Willebrand factor here. As you can see here, the ratio of liver versus adipose tissue is far high in cirrhosis compared to healthy. In von Willebrand factor, that ratio is not that significant. And what about the ratio of cirrhosis versus healthy? That is what this graph shows. Factor 8 in adipose tissue, factor 8 in liver, von Willebrand factor in adipose tissue. This is remember that this is the ratio between cirrhosis versus healthy people. And we also looked into the ADAMTS13. ADAMTS13 is a protein which is going to chew down the von Willebrand factor. So these protease levels are also high in adipose tissue compared to liver when you take the cirrhosis versus healthy ratios. So now the question will be here, does the cells which produce factor 8 or the factor is produced by adipose tissue is functional? Because as you all know, there can be different isoforms of the same protein which is expressed in different tissues. So what if the isoform or the factor 8 which is produced in adipose tissue is not functional? How to prove this? So what we did is a simple experiment. Inject these cells, these adipose tissue stromal cells or mesenchymal cells, into a hemophilic mouse. But there is a problem. If you inject human cells into a mouse, what will happen? It's a xenotransplant. It will be rejected. What is the solution? Inject the cells into an immunocompromised mouse. That's what exactly we did. We mated, we crossed a factor 8 deficient, factor 8 knockout hemophilia mouse with a Nord-Skid mouse. This Nord-Skid mouse is immunocompromised animal. And we looked for those animals which had both mutants together. That means these animals will be deficient for hemophilia for a factor 8 and they will also be immunocompromised. To those animals, so we verified those animals by doing specifically looking for those knockout of those genes. That is that these PCRs are showing that. So now the human adipose tissue stromal cells were injected into the mouse, which is a double knockout, which is knockout for hemophilia factor as well as the. So after that, about 10 days, we killed the animals and sectioned the liver for a microscopy. This is what we found. This F480 is a well-established marker for macrophages or Kupffer cells in mice. You can see those green spots. It may not be very clear in this because of the background lighting. So you can see, sir is not coming here, but I'm trying to, yeah. See, these green cells are the Kupffer cells, and you can see this nucleus of hepatocytes. These are hepatocytes on the background. And as you can see here, and here, these are the factor 8 positive cells. So you can see after injection with those.
Can you decrease the lights, please, so that
those adipose tissue derived cells, you can see factor 8 positive cells which in the liver of these animals. This blue color is the nucleus of these cells, which are stained for DAPI, and you can see among these cells, you can see this factor 8 positive cells. That means these factor 8 positive cells are deposited in liver. At least they are surviving there, at least on the 10th day. But still the question remains unanswered whether this factor 8 is functional or not. So from this animal we collected. So now this collected blood and did a factor 8 functional assay. So what is a factor 8 functional assay? So Western blot is not a functional assay. It is a qualitative and semi-quantitative assay. So it does not tell whether the protein is there, it will tell, but the protein is functional or not, it will not tell you. So we did a functional assay. Here, you remember the blood coagulation cascade. It's complex, several proteins are involved, but all you have to remember is here. This factor 10 has to be activated to factor 10A, activated factor 10A. And you need all these components for the activation of that. And activated factor 8 is a key component in this reaction. So essentially, you supply everything for this pathway to run, except the only one thing will be deficient, that is factor 8. So now the entire reaction is dependent on the factor that you are going to supply from the mouse blood. So now if factor 8 is present, the factor 8 is bound to factor 10A is going to, factor 10A is essentially a protease, as you all know. Dr. Ankur was just describing about dabigatran and all those things. So factor 8 is going to, factor 10A is going to cleave this compound, because this has exactly those peptides which are present in thrombin, in prothrombin. So factor 10A will release this fluorophore. This compound is conjugated to the fluorophore, and you will get a signal. And that is how the factor 8 is measured here. So we found that, yeah, the factor 8 is produced by adipose tissue is functional. And now what we did is that we killed these animals after 6 months and looked for factor 8 assay. And of course, this is the graph which we got. And as you can see here, the animals are producing factor 8 and functional factor 8. But there's a problem. When we looked into the liver of these animals after 6 months, we couldn't find a single surviving adipose tissue derived cell. All were dead. So from where the factor 8 is coming now? That was the question. So what we did is that each organ from this killed animal and looked for factor 8 mRNA. And interestingly we found there is no factor 8 is produced in liver, no factor is there in lungs, but the only tissue from where we were getting this factor 8 signal was adipose tissue. So it is like the mouse story. The mouse wanted to become a princess. So similarly, wherever, even if you send these mesenchymal cells which are derived from adipose tissue, finally they will not reside in liver. In liver they are going to die, and they will only survive from where they came from, that is the adipose tissue. So the signal was coming from adipose tissue. Now we understand that the stromal cells which are transplanted survives only in adipose tissue, and where they are producing factor 8. And now the question is that, what are these mesenchymal stem cells? So it's a complex question. In fact, there are certain markers for mesenchymal stem cells, but there's a huge disagreement regarding many of these markers. So what we have to prove is that we have to really find the identity of these cells. So far our studies shows that mostly these cells are towards the pericytes. Pericytes are those cells which are found around the vasculature. So these cells are derived from that. Western blots which we done to confirm the identity. But there's a problem with Western blot or immunofluorescence like this, because we are all, whenever you do an immunofluorescence or a fax or a Western blot, you already have an idea about, you already have a bias regarding your search, what you are searching for. You are always biased. So this question can only be answered by a single cell sequencing. And that is what we are trying to do now. So now some practical thoughts. What are the clinical implications? We all know that obese people are more prone to stroke and thromboembolic events because of increased factor VIII production. Can it be because of the factor VIII produced in adipose tissue? Because you know, more obese, more adipose tissue, more factor VIII. Cirrhosis is a prothrombotic condition. What is the contribution from adipose tissue? And now comes cell therapy. We found that adipose tissue MSCs or mesenchymal stem cells produce factor VIII. So they should be administered with caution in prothrombotic states. So for example, as a patient, if you are trying to save a patient who suffered from stroke by injecting MSCs, probably you are not doing the right thing. And you may remember the NEGM gene therapy paper, where they tried to express factor VIII in CD68 positive cells. Probably that is not the right thing to do. You should probably target mesenchymal stem cells in hemophilia gene therapy. That may be a better target. And now I'm coming to the part 4, which is the mechanisms. Science forms a hierarchy. Physics rests on mathematics, chemistry on physics, biology on chemistry, and social science on biology. Luckily, there are no social scientists here. Deeper you go, more fundamental the mechanisms become. So these are the identity of the adipose tissue stromal cells. As you can see here, these are the adipose tissue stromal cells from a healthy person, and this is from the cirrhosis patient, a cirrhosis patient. See the nature of these cells. Even in the first glance, you know they are different. They are very different. And morphologically they are different, but also they are functionally different. The mesenchymal stromal cells from cirrhosis patients proliferate faster. So you should think about the x-axis and y-axis. The y-axis is in the log of 2, or it's binary. It's a binary axis. So you can see that the mesenchymal stem cells from adipose tissue are proliferating much faster than those cells from a cirrhosis patient. And to verify it further, we looked for PCNA, which is proliferating nuclear antigen, and that's a proof that they are more in mesenchymal cells from cirrhosis patients. And same is true, and also we looked for Ki67, which is also a marker for proliferation. As you can see here, that's also high in patients with cirrhosis. And further, we also looked into cyclin D1, which is a cell cycle related. So more cyclin D1, more cell division. So that's also proof that the mesenchymal stem cells from cirrhosis patients are proliferating faster. And again, what is the functional? So you told about the markers of mesenchymal stem cells, but we are not sure. Show by their fundamental properties. So the definition of mesenchymal stem cell is that you know that mesenchyme can give rise to 3 lineage, which are bone, adipose tissue, and cartilage. So we differentiated the mesenchymal stem cells from adipose tissue derived from healthy people and cirrhosis patients into osteocytes first. So this is a Wongosa stain. When cells are differentiated into bone or osteocytes, they accumulate calcium. And silver in the Wongosa stain is deposited on those calcium-rich areas. So as you can see here, the mesenchymal stem cells from a healthy person, or those gave rise to more bone formation than mesenchymal stem cell from a cirrhosis person. That means their efficiency to differentiate, the efficiency of mesenchymal stem cells from a healthy person to differentiate to bone is far better than those collected from the mesenchymal stem cells from a cirrhosis patients. So the same is true for chondrocyte. Here we try differentiation into chondrocyte. This blue stain stains for the glycosaminoglycans. As you can see here, the healthy cell, the stromal cells from healthy differentiated far better than cells from cirrhosis. Same is true for adipocytes. So you can see here the red color. This is oil red O, which stains the fat inside the mesenchymal stromal cells. So again, so in short, the differentiation capacity, in other words, that refers to the youngness of the stromal cells in healthy is far better than cirrhosis. Or the stromal cells are in a healthy person is biologically younger than cells from a cirrhosis person. So again we observed that although these cells, stromal cells from adipose tissue from cirrhosis patient proliferate fast, they undergo senescence also very fast. So how we know that? This is a beta-gal stain. So beta-gal is a marker of senescence. If senescent cells are there, if senescence is there, these cells will stain blue, as you can see here. This is from cirrhosis. See, these cells are stained deeply blue compared to the cells which are from a healthy person. Which means that although the mesenchymal stromal cells proliferate fast, they also undergo senescence fast. So cirrhosis harms you, harms not only the liver, but also almost all other organs. They induce premature senescence. And now there are several theories for senescence. So we wanted to look into that. So one theory is that telomere shortening. As cells proliferate, the telomeres shorten. The second theory says that the oxidant stress and free radicals impact damage the DNA and affects damage the DNA and proteins and cause senescence. So both are true in cirrhosis. So we looked into the oxidant stress imparted by the free radicals on various proteins. Those proteins which have the disulfide bond and tyrosine residues are more prone to oxidation. So what we did is next is that we looked for those proteins which are differentially oxidized in cirrhosis in adipose tissue. And what we found is that we got a lot of proteins. And one protein we were particularly interested was PTPN14. And what is the function of PTPN14? Phosphatase. That protein is a phosphatase. A phosphatase is a protein which removes the phosphate group from a phosphated protein. When proteins get phosphated, when they are getting activated. You know that when cell signaling pathways are active, many kinases add a phosphate group to many critical proteins, and that decides the activity of these proteins. So when a phosphatase is oxidized, it loses its activity. When it loses its activity, phosphated proteins, the level of phosphated proteins, that means the level of activated proteins, are up. Indirectly, I am telling it will simulate the increased activity of the kinases. I know the time is so we did a Western blots. It's not just a matter of some kind of mass spec, but we validated it by Western blot meticulously, not only those specific proteins, but also the downstream proteins one by one, like J and K. The TCP/TP is a protein which is it's a phosphatase, and we looked for the specific oxidate form of TCTP, and which is high. And we looked into the downstream signaling like JNK, phosphated JNK, PAKT, and PSTAT1, STAT3, and phosphorylated STAT3, and CMYC, which comes in the downstream. And we found that, yeah, all these are upregulated. That means the tissue is, the entire adipose tissue is going to a pro-proliferative or pro-angiogenic state. And we also confirmed that where we didn't have antibodies, we confirmed by real-time PCR like MMP1, MMP2, and MMP9, and all those things. And I don't want to bore you into the details. This is the proposed mechanism where the cirrhosis generates a lot of reactive oxygen species because a lot of metabolites are getting accumulated, and the liver continuously tries to, in cirrhosis, the liver always tries to regenerate because it has a best regeneration potential, but it is never achieved. That's a futile regeneration. In this process, it also flushes out a lot of cytokines into the bloodstream. They're all organs and all tissues are cooked in this. So adipose tissue is not an exception for that. So that creates a pro-angiogenic, and actually under stress, you emulate your childhood or your embryonic state. As I was showing you the picture of the congenital rubella child. So a stress situation. So you are going back to your ancestral states. So that induces a pro-angiogenic state in the adipose tissue. In the public health importance, but I don't think I have enough time. I'll just say, knowledge, everybody worked in my lab in last 10 years, and I especially thank Dr. Pamacha and Dr. Kumaraswamy for providing me the tissues, because they started this work. At that time, I got the adipose tissue samples from them, and of course, I thank ILBS for providing me a space, and yeah, thank you everybody for the